Hello everyone. Now we are going to discuss about the chronic inflammation. Chronic inflammation is a response of prolonged duration in which inflammation along with tissue injury and repair coexist at varying combination. What are the causes of chronic inflammation? First is persistent infection with microorganism. Some of the microorganisms are difficult to eradicate and may result in chronic inflammation. For example, mycobacterium. Hypersensitivity reaction can also result into the chronic inflammation. Prolonged exposure to potentially toxic agent like potentially toxic agent, they can be endogenous or exogenous. In endogenous, cholesterol deposition, which is seen in atherosclerosis, and in exogenous toxic agent, silica particle can also be responsible for the chronic inflammation. So broadly we have seen three causes: persistent infection, hypersensitivity reaction, and prolonged exposure to potentially toxic agent, which can be endogenous or exogenous. Now we will discuss about the morphologic features of the chronic inflammation. Characteristic feature of chronic inflammations are inflammation with mononuclear cells, namely macrophages, plasma cells, and lymphocytes, along with the tissue destruction. Tissue destruction is a feature of chronic inflammation. This can be due to the offending agent or by the inflammatory process itself. And there will be attempts at healing. Body tries to heal the injury via angiogenesis and fibrosis. So these are the three morphological features of chronic inflammation: mononuclear cells, tissue destruction, or attempts at healing. Cells and mediators involved in chronic inflammations, as we have discussed, primarily the mononuclear cells play an important role in chronic inflammation. Now we will discuss them one by one. Macrophages. Macrophages are the dominant cell in chronic inflammation. Macrophages, how they produce? In bone marrow, by hematopoietic stem cells, there is a production of monocyte, which ultimately release into the blood, and these monocytes get activated into the macrophages during inflammation or various processes. On the other hand, yolk sac and fetal liver contain some progenital cells, and they form the tissue resident macrophages like Kupffer cells in liver, alveolar macrophages in lungs, and microglial cells in CNS. Half-life of the macrophage. Half-life of blood monocyte is short, normally 1 day, but half-life of these tissue macrophages can vary from months to year. Products of activated macrophages, once get activated, they eliminate injurious agent, play an important role in repair, and may result into tissue injury. One of the very important topic is activation of macrophages. This plays a very important role in inflammation and healing process. So we will see how macrophages get activated. There are two pathways Classically activated macrophages known as M1, alternatively activated macrophages known as M2. So, microbial products like endotoxin or interferon gamma secreted by lymphocytes will result into the activation of classically activated macrophages M1. These classically activated macrophages secrete or produce reactive oxygen species, nitric oxide, or lysosomal enzymes, and will responsible for the microbicidal actions, phagocytosis, killing of bacteria and fungi. And on the other hand, they secrete some cytokines like interleukin 1, interleukin 12, interleukin 23 and chemokines, and further play an important role in inflammation. Now we see the alternatively activated macrophages. Cytokines like interleukin 13, interleukin 4 and interferon gamma, they activate macrophage into the alternatively activated macrophage M2. These M2 type of macrophages secretes growth factors and TGF beta, which plays an important role in tissue repair and fibrosis, and they secrete some cytokines like interleukin 10 and tissue growth factor beta. And as we know, interleukin 10 and tissue growth factor beta has an anti-inflammatory effects. So M2 macrophages has important role in tissue repair and fibrosis, and they have an anti-inflammatory activity. Other cell types in chronic inflammation that play an important role is lymphocytes. Microbes and other antigen activate T and B lymphocyte by various mechanisms, and they play an important role in chronic inflammation. CD4 T cell, known as helper T cell, once activated, they have three classes: Th1 cell, Th2 cell and Th17 cell. H stands for helper T cell. Helper T cell 1 secrete interferon gamma, which activate macrophages by classical pathway. As we have already discussed about the action of classically activated macrophages earlier. Th2 cell secrete interleukin 4, interleukin 5 and interleukin 13. They play an important role in recruitment of the eosinophil. They activate macrophages by alternatively activating pathway. Third type of CD4 T cell is Th17 cell. They secrete chemokines and responsible for the recruitment of neutrophil and monocytes. This is a diagrammatic representation of macrophage lymphocyte interaction in chronic inflammation. Antigen presenting cell react with the T helper cell, CD4 cell, and after activation CD4 cell secrete interferon gamma, which convert monocyte into the activated macrophages by classical pathway. And these activated macrophages secrete interleukin 12, which further activate Th1 cell, and they keep complementing each other's function. And activated macrophages may result into the formation of the granuloma. So this is a diagrammatic representation of a granuloma, which we will discuss later on. Another cell type is eosinophil. Eosinophil is particularly important in allergies or parasitic infection. Eosinophil has major basic protein, a cationic protein, which is toxic to parasitic infections. Mast cell. They are widely distributed in connective tissues and participate in both acute as well as chronic inflammation. Mast cell secretes variety of cytokines. Mast cell, they bind with the Fc portion of the IgE immunoglobulin and release mediators like histamine and prostaglandins and take part in inflammation or responsible for the development of inflammation. Neutrophils. In chronic bacterial infection like osteomyelitis, neutrophil can be a predominant cell. Now we will discuss about the granuloma or granulomatous inflammation. Granulomatous inflammation, one of the very important topic. Granulomatous inflammation is a form of chronic inflammation. It is a type of chronic inflammation. Granuloma formation is a cellular attempt to contain an offending agent that is difficult to eradicate, which cannot be eradicated so easily. So it is an attempt to contain that offending agent, like seen in Mycobacterium tuberculosis, which is difficult to eradicate. Normally, granulomatous inflammation associated with the normal tissue injury or injury to the adjacent normal tissue. How granuloma develop? Activated macrophages has abundant cytoplasm and they resemble like an epithelial cells. That's why they known as epithelioid cells. Activated macrophages may fuse to form a multinucleate cell which is known as giant cell. Granuloma can be of two types: foreign body granulomas or immune granulomas. Foreign body granuloma is cited by inert foreign bodies which result into the inflammation. There is absence of T cell mediated immune responses. On the other hand, immune granulomas, variety of agent causes persistent T cell mediated immune response and they are difficult to eradicate. T helpers lymphocyte get activated by the antigen presenting cell and secrete interferon gamma. These interferon gamma activate macrophages by classical pathway into M1 macrophages. In some parasitic infection, helper 2 cell, TS2 cell, also responsible for the development of granuloma, which is seen in schistosomiasis. Now we will see the morphology of granuloma. Here we are taking example of caseating granuloma, which is seen in Mycobacterium tuberculosis, a center portion of necrosis surrounded by epithelioid cells, epithelial like activated macrophages, epithelioid cells with the collar of lymphocytes and fibroblast and giant cells are also noted at places. The central portion of necrosis is because of the hypoxia and reactive oxygen species. Examples of granuloma, one of the very famous example is tuberculosis, leprosy. Granulomas can be seen in syphilis, cat scratch disease, sarcoidosis, and Crohn's disease, which is a form of inflammatory bowel disease.